everybody, welcome to this revision session for Developmental and Evolutionary Genetics. I am Dr. Mohammed Ahmed. I wanted to make this short video prior to our online revision session that we will have, so it gives you the opportunity to have a listen over, think about, and write down any questions you may have during that session. So it becomes more of an interactive session than it does me just talking to you talking to you directly. So definitely have a listen to this. Any questions you have, put them down and we'll dig into the sessions. Before I actually get into the actual revision session, I want to highlight that there are some slight changes to the examination, but please do keep checking in the Now module page. Some of these criteria may change over time, so the most up-to-date information is available on the Now module page and in correspondence from course leaders. And if you're not sure, just ask me directly and I'll try to find out for you. So the exam format hasn't changed at all. Still answer 3 questions in total. So you have one section A. So you have section A, section B, section C. You have to answer 1 question from section A, 1 from section B, 1 from section C. And each section has 2 questions. So you can answer 1 in each section. All questions carry equal marks. You should submit all your answers into one single document. If you're doing diagrams, you cannot copy and paste from the internet. You will have to hand draw them and put them in by scanning or picture taking through the phone. If you have an NEC or a statement of access, you will get extra time and this information will be available in due course. You will have 24-hour window to access this paper. As soon as you access it, the timer will start for 4 hours. As it is online this year, you will have 4 hours for the examination to complete and submit. So please submit the answers prior to the 4 hours being completed. Some additional information which changed this year: you're only allowed to have 1,000 words per question. So for example, you answer question 1 for section A, you have 1,000 words for that. If you answer question 1 in section B, you have 1,000 words for that. And if you answer one question 1 for section C, you have 1,000 words for that. Please note that we will actually put your answers through Turnitin, so don't be tempted to cheat or plagiarize anything. And again, going back to the total number of hours, you'll have 4 hours from the beginning of the exam and 24 hours access. Please keep your eye on the Now module page and your emails and the Now module, Now personal exam timetable as well. These should not change. Exam timetable should not change in any capacity. But please do keep checking your emails on Now module page for any additional information that you may require. And again, you are welcome to email me any questions directly. So let's just dive into the revision session. Again, I'm not going to cover all topics, but I just want to give you a feel of what you could expect to see and what kind of questions I've covered in the past. I've provided 3 exam questions this time. And I'll give you an idea that some questions can be actually answered by multiple same sources of the multiple kind. And I'll explain what I mean by that in a few seconds. So the themes that we covered this year were quite wide-ranging, especially in embryonic development. We looked at induction and competence. We looked at access to term development and early embryo or limb development, positional identity, looking at Hox genes, developmental systems. We did actually look at neural tests this year, but I think it was online. If not, ignore that. And we also looked at FCF, sonic hedgehog signaling to digit formation. So I want to give you these 2 past questions we've had in the past 2 or 3 years. The first question: explain vertebral limb development from start to finish, talking about digit formation, including genes involved. And the second question, evaluate how mutations that disrupt embryogenesis inform our understanding of normal development. Please give examples. So straight away you notice that the questions are based on developmental biology and are asking for examples. So just be aware of that. That if a question comes like this, what is the kind of topics I should be referring to? At the end, it will also ask for examples. So just be aware that you have some examples at hand to provide for this. The really interesting thing about developmental biology that I taught this year to you is that if either these questions come up or a combination of these questions come up, you can really amalgamate a lot of information together to answer multiple questions. So I just want to give you a kind of a flavor for that today. So these two questions both come from the kind of developmental aspect, looking at derivative of tissue, looking at blastocyst from the beginning, producing inner cell mass, and the epiblast, and then they go on to make these three germ layers known as embryonic ectoderm, embryonic endoderm, embryonic mesoderm. And this is something you could mention very briefly in the introduction of your essay, which I'll break down in terms of the structure of an essay in my questions, answering my questions. It should be like or similar to that. Gastrulation, so what is the definition of it? It's not birth, marriage or death, but this process which is truly important time in your life. This is cells on the surface of the embryo moving into the more interior location and the kind of processing and development and gene regulation that comes about with it. Some of that gene regulation that we're referring to is just looking at the compaction, cell division that occurs during embryogenesis, which is essential for the normal embryo to develop. So we can mention some of these processes, some of these terminology and what they are before you go into the actual essay itself. We also cover three axes of development: dorsal to ventral, anterior to posterior, left and right, very little, but some of it. And this could be applied to digit formation and also embryogenesis in general. And we could apply all this kind of notes and knowledge that we've gained over the last year and apply to both questions at hand. I like this summary because it shows very nicely the different pathways involved in limb development in particular. So you have the kind of interaction between sonic hedgehog from the posterior and FGF to the distal, and proliferation and migration in both axes. And then you want to perform or create the posterior to the distal version axis. You have the Wnt signaling of Wnt7a and sonic hedgehog. And this diagram will be a good way of explaining, if this question comes up in the examination, to explain what the three axes that are being formed and how these key signaling pathways play a role in that. And don't forget, not just by describing it, you need to explain to me exactly what sonic hedgehog is doing, which I'll go through in the next few slides, what I mean by that, and how Wnt plays its role in this axis formation, and what kind of cell populations are involved in there. And again, I'll touch on this as I go through the presentation. Hox gene is a good one. Could we add Hox genes to these two questions? The expression of Hox genes, what is the attributes, why they're so important, and it's really about the positional identity and how position identity is determined by the Hox expression in different locations, and how we could apply this knowledge to the actual two exam questions that we've been proposed. So maybe we could actually add the Hox genes, not much of it actually, but a little bit of it, just to show that we know that Hox genes are also important in the three axis or digit formation that we're referring to. I like this diagram also because it kind of illustrates very nicely the different axes you can have forming. Again, this could be really interesting for especially the first question that we've got, is that looking at digit formation and the different three axes that are involved in causing or leading to a digit formation correctly. So this diagram we could use for understanding, but also in the essay just to describe the three main axis formation. And again, there's more details in my lectures and the lecture captures that I've recorded in the past. We do have a look at that. And again, most of my information diagrams are cited, so you can also have a look at that information for further reading. So limb development, again, looking at both questions, what do I need to talk about? I need to talk about formation of the limb bud, how does that work? From initiation to actual final digit formation. You need to talk about generation of the proximal distal axis, the anterior, posterior, dorsal, ventral, and this goes back to that diagram I just showed you earlier on. How do these three axes work together, and how do they form digits and joints and form cartilage? Examples of gene regulation. So we looked at sonic hedgehog, FGF signaling. We've looked at Wnt, but also the other transcription factors such as TBX5 involved in transcription of forelimbs, for example, and you've got TBX4, which are hind limbs. You can mention these quite easily. Don't forget also you need to mention these key zones of interest when you're looking at embryogenesis and digit formation. You have the areas of ZPA, zone of polarizing activity, the progressive zone, and you've got the AER. And this is quite important for the initiation of limb formation. And again, why certain sites are more predisposed to limb development rather than digit formation happening in every site of every cell in the body. Certain localized factors are involved in that, right? So the AERs, or the apical ectodermal ridge, what is it? What does it represent? How is it known to be interesting or known to be important in limb bud formation, digit formation? So the zonal polarizing activity, again, using experimental data where we can take away this segment of cells and replant them in a different part of the developing limb bud, and we have the kind of mirror image of resulting skeletal pattern which mirrors each other. And the ZPA expresses high levels of sonic hedgehog. Again, if this question comes up, it's something that you can mention in terms of digit formation. Sonic hedgehog itself can be described in this pathway. I don't want you to put in every single detail for the sonic hedgehog pathway, please. I just want you to show me the key main steps. So you're looking at sonic hedgehog activation, binding to its receptor known as patch or PTC, and the cleavage and activation of three different GLI proteins, GLI1, 2, and 3. And pretty much that's it. And this diagram might be helpful, especially panel B will be helpful to draw that. I would avoid panel A altogether because I don't see it helping you in any capacity. But panel B is quite nicely demonstrate the effects of sonic hedgehog on digit formation. Again, now moving on to slightly the second half of the question when it talks about examples. So which mutations or how do we know sonic hedgehog is important for limb development is through mutations in animal models or studies done in humans where they found that certain mutations, certain transcription factors are responsible for that defect. So GLI3 mutants are polydactylous, we have additional digits, and this shows that is important, while the other two GLI proteins, 1 and 2, don't show any phenotype at all. Again, we will show you some additional diagrams where you can be very interested to show part of your understanding where you have mechanisms of dorsal ventral patterning and AER positioning. So you have high expression of S7A in the dorsal, where you have the N1 expressed in the ventral, which kind of has this balance between expression of AER formation and dorsal patterning. And this pathway could be really important for you to describe what's going on. Also, always provide human or mouse analysis or examples in terms of mutations or disease states part of your questions, because that really shows you real understanding of not just the pathways and just describing them, explaining them, but also shows the real life implications of when these genes are defective. So in this case, I've mentioned polydactyly and how sonic hedgehog can be part of that, for example. And also examples of GLI3 mutations and sonic hedgehog, but also using type 1A and type B mutations. For example, what would happen if Wnt7A is mutated? What kind of effect would that have on digit formation? So I also want to give you a third question, which gives you a completely different kind of vibe in terms of the topic you need to look at. So many of you did this topic as your oral presentation, which will give you a bit of advantage in terms of looking at this. So if this does come up, you will have hopefully a better you will have a more of a grasp of what needs to be done. So the question that came up in the past was discuss the importance of stem cells and their role in regenerative medicine. Please provide examples. So instead of going through the whole lecture again, I thought it would be easy just to break down what topics we should look at. I've used the first learning outcome slide from the lecture because this is actually a perfect way of looking at the essay and the question itself. So what you need to do is you need to describe and discuss stem cells and their properties. You need to discuss embryonic stem cells. You need to discuss the role of adult stem cells and the mechanism of tissue regeneration. Maybe mention even cancer stem cells in this concept. It'll be interesting to see where you would put this. And also really important looking at induced polypotent stem cells and therapeutic use of stem cells. So all of this comes together in terms of how you would answer the question. My next line is just going to tell you how to break up all the essays in this module, especially if you're writing for me, how you should go about writing that in terms of a structure for the essays. How many of you just remember this from the last year in our year 2 module together? So, for example, discuss the importance of stem cells and the role in regenerative medicine. Please provide examples. So you've got two parts to this question. You've got to discuss the importance of stem cells, so why they're important, and their role in regenerative medicine. So you've got two parts there, but also you've got a third part which says you have to provide examples. So the provide examples only comes into this part B in their role in regenerative medicine. So give examples of where stem cells have been used for regenerative medicine. So in introduction, we'll describe and discuss stem cells and the properties and the characteristics, the different types of stem cells, what's so special about polypotent, what's totipotent, all these other aspects of definition in the lecture. The main body of the essay will then move on to describing and discussing embryonic stem cells. Again, you can put in the developmental aspects of looking at 3 germ layers and part of that because embryonic stem cells will become them and actually produce all 3 particular cells populations. Discuss the role of adult stem cells and the mechanism of tissue regeneration. Do you know the difference between adult stem cells and embryonic stem cells? Discuss cancer stem cells and what are they? Why are they important? So where would this go in this essay? Would you include it in this essay? It's one of those important things that you need to think of. And then you're talking about the regenerative role here, the second part, the role in regenerative medicine. Discuss induced polypotent stem cells. What are they? So what are induced polypotent? What's the mechanism of it? How can they be used? Are there advantages of using induced polypotency? And are there disadvantages of using polypotency, which will show you the really great understanding of what iPS stem cells do, which kind of treatments are being used. So this is the last bit, discuss therapy use for stem cells. So you can talk about embryonic stem cells, you can talk about iPS stem cells, you can look at somatic nuclear transfer for treating diabetes, an example as well. So don't forget all of these examples is quite nicely laid out in the lecture for stem cells and regenerative medicine. Treating Parkinson's, Alzheimer's, and again, examples can be used freely and widely and doesn't have to be restricted to what I've provided into your thing. And if you want to borrow maybe one of your friends' presentations who did the stem cell presentations, you're more than welcome to do that as well, as a point of reference and revision. And the conclusion is take-home message. So what's so important about the aspects of stem cell regenerative medicine? Why is it so important? Why is it so interesting for us to know these principles and these characteristics of stem cells for treating disease? And that's the way I would break down every essay. So you have 3 main components. You have the introduction, you have the main body of the essay, and you have the conclusion. And make sure you hit all of these points as you go through. Of course, every point is different for every essay question, but in generic fundamental essay writing, these are the 3 core subtopics. Make sure you understand the question that's being proposed. Is it asking for one aspect, 2 aspects, and is there a third element that you need to consider as well? So in this case, discuss the importance of stem cells, part A, and then the role of regenerative medicine, part B, and the third element is you have to provide examples, and this is what this example is going to do. Okay? So thank you very much. Thank you very much for listening, and I look forward to seeing you all on the next few days, in a few days' time. And please listen to this, put your questions in writing, and bring it to the session. And hopefully, we can answer. I can answer all those questions, and hopefully, your exams go extremely well. Thank you very much. I'll see you soon.